this morning is Marina Strocchi of Imperial College and King's College London. And Marina's presentation title is Cell-to-Whole Organ Global Sensitivity Analysis and Parameter Inference on a 4-chamber heart electromechanics model using Gaussian process emulators.
Thank you. It's a pleasure to be here to present my work. So I would like to start thanking everyone who contributed to this work, everyone at Imperial and all my external collaborators from Gernot's group who provides us with CARP to run our simulations, the statisticians who provide support with all the stats in the work, and the clinicians who provide the clinical data. So I hope I don't need to convince anyone here that the heart is complex. We have processes going from single proteins to the cell, to the tissue and whole organ and body with the circulatory system. When we look at cardiac diseases, they can hit any of the scales. However, when we look at what cardiologists can measure, very often this is restricted to whole organ scale. And for them, it's quite difficult to link what's causing the symptoms, so to understand what's going on. When we look at treatment, we have a lot of drugs or devices available, and it's quite difficult to understand what's the optimal treatment for a specific patient. So what computational modeling can do is provide mechanistic insights by linking all these different scales to clinically measurable outputs. And we can also help select the optimal treatment for a patient by building a patient-specific model and then testing in silico different treatment modalities and select the optimal one. So hopefully today I will convince you that computational modeling can do both. We start with an anatomical model generation, so we can now do this from CT and MRIs. We have a fairly robust pipeline where we post-process the segmentation, we smooth it, and we mash it with linear tetrahedra. We assign fibers in the ventricles and in the atria. We use the Bayesian rule-based method in the ventricles to represent transmurality, and the universal electric coordinates to map an ex vivo diffusion tensor MRI on the atria. And we also have a way to define universal coordinates in the ventricles, the left atrium and the right atrium, and this helps us defining fast conducting regions in the atrium and the ventricles, and also to define boundary conditions. In terms of computational framework, it's a fairly complex one. It's an electromechanics simulation. So we have the single cell scale represented with a ToR-ORd-Land model and the Courtemanche model in the ventricles and the atria. And these are coupled with the Land human model for of contraction for ventricles and atrial cells through the intracellular calcium transients. So as quite a few of you know, these models are ODEs based, but they're quite complex and they have a lot of parameters. In this case, we have about 50 for each cell model. We represent tissue electrophysiology with the reaction economic model, and Gernot has presented this earlier. We solve for the local activation time in the geometry over the space. We have as parameters the conduction velocities in different tissues, and for this patient, we know the location where the stimuli start because this patient had a pacemaker, so we know the location of the right atrial and the right ventricular leads, so those are not unknown, but we do not know the atrioventricular delay, so that is a parameter in the model. For the passive mechanics, we have a Gaussian model, which is a transversal isotropic representation of the myocardium, of the ventricles and the atria. We have separate parameters for the ventricles and in the atria, so you can see how this is getting quite complicated. We have the passive stress summed to the active stress in the fiber direction, and this gives a total stress of the system. And we put this into the Cauchy equation of motion to solve for the local displacement over time, so basically the heart contraction, given, of course, boundary conditions. So we have a representation of the pericardium with normal springs. The stiffness of the springs is a parameter in the model, and it varies over space. And we also have a representation of the circulatory system based on CircAdapt. So here we have the CircAdapt model coupled with the 3D model, 3D PD model, by basically adding an equation to the finite element solve so that the volume estimated in the PD and the volume estimated in the OD match up, and this gives us the pressure in the 4 chambers. And also this circulatory model is quite complex with quite a few parameters. So given the number of parameters I've been listing and the complexity of the simulations, doing a sensitivity analysis is pretty much impossible because the simulations cost 4 or 5 hours on 512 cores, which is an insane amount of compute time. And so we need to be smarter, so we use emulators. So for every output we want to fit, we substitute the expensive simulator with the train emulator, and we do this also to run the sensitivity analysis. So we start with the coarse sampling of the parameter space, we run expensive simulations, we extract the outputs we want, and then we train the emulators, and we look at the performance of the emulators. So this is, I think, quite a nice picture to show how well the emulators are doing. So in red you have the observations that you're trying to match, and in black you have the predictions, and you want them to be close, of course, but we also have the emulator uncertainty. So you see that even if sometimes the predictions are not that close to the observations, the uncertainty of the emulator is still able to account for the observations. So we use these trained emulators to do a global sensitivity analysis. So I'm not gonna go into details about this because this is published, so if you're interested in the publication, it's at the QR code. But basically what we were able to do was to have a lot of parameters representing different processes at the cellular, tissue, and whole organ and circulatory system scale, and we link them to clinically measurable outputs for all 4 chambers on the y-axis. And so this provides basically a link between all the processes we considered with clinically measurable outputs, which is pretty much impossible to do clinically. So the second part of my talk is very new results. It's results I generated pretty much last week. So it's about parameter inference, this is obviously a very complex problem, and again we tackle this with emulators. So all the work I'm presenting here is done with Historia, which is a library implemented by Stefano Lombardi, who was a former member of the group. And basically, history matching is a technique where we try to restrict the parameter ranges to areas where the outputs are consistent with the clinical data we have available. So on one side, we have the measurements with the target value and an uncertainty associated with them, which is quite important to think about. And on the other side, we have the emulators with their prediction and the uncertainty associated with the prediction. And for every sample in the parameter space, we compute an implausibility measure I, which has at the top the difference between the prediction and the target, which obviously we want to be small, but at the bottom we also have the uncertainty of the emulators and the uncertainty associated with the data. And then we can define a cutoff on the implausibility measure and divide the samples between not implausible and implausible. Not implausible means that at this iteration, the prediction is close enough to the target value, while implausible it means that it's not. And history matching done with the emulators is an iterative procedure where we refine the emulators at each step so that the uncertainty on the emulator goes to 0, or at least it's a lot smaller compared to the uncertainty on the data, so that when we define the cutoff on the plausibility measure, we can say we are a cutoff number of standard deviations away from the target value. So to define our targets, we have clinical measurements. For this patient, we're quite lucky because we had invasive pressure measurements. So this patient was selected for CRT upgrade from right ventricular pacing to biventricular pacing, which means that during the upgrade procedure, this patient underwent left ventricular invasive pressure measurement. And we had quite a lot of them. And here you can see a typical pressure trace with the red beats highlighted representing non-ectopic beats, which we used for this analysis. And basically this resulted in 30 usable beats. In blue there you have the mean pressure trace with the one standard deviation has 3 standard deviations, and basically what this gives us is a physiological bit-to-bit variability, and therefore we have a mean and uncertainty associated with each feature. We're trying to fit, in this case, the maximum pressure and the end diastolic pressure. For this patient, we also have an ECG-gated CT, which means that we can also extract motion, and then we can deform the mesh and extract volume transients. We can then synchronize the left ventricular volume transient with the pressure trace to get the pressure volume loop, and this helps us define uncertainty on the volumes as well, which otherwise we wouldn't really have. And this provides us with target values for volumes of the left ventricle, right ventricle, and the left atrium as well. So for the fitting or the parameter inference, we start with the sensitivity analysis for the outputs we're trying to fit. We can then select the important parameters for each output, and you can see there are not that many. We then perform history matching, and then what we can do is to repeat the sensitivity analysis, restricting the parameter ranges of those parameters we are trying to infer. And as you can see, the parameter ranges get restricted quite a lot. And so because the GSA is dependent on the parameter range, these parameters disappear. Now they're not important anymore. And then we can go around again with this new sensitivity analysis, select different important parameters, perform history matching again, restrict GSA. And we can go around as many times as we want. I did this 4 times. And at this point, the outputs are within physiological ranges. So this is what it looks like. So each plot is a different output I'm trying to fit. The gray areas are the uncertainty ranges for the outputs, and the dots are different simulations. So the blue is where I started, which is all over the place. And as we proceed in the history matching, you can see that the dots fall within the shaded area, which is what we want. So this is quite promising preliminary results to perform parameter inference on such a complex model. So the last thing I want to touch on is model validation. So as we had a lot today, yesterday, this is very important, and if we want to move towards clinical translation, this is very fundamental. And so what we're doing now is that we're running our clinical trial, where we have atrial fibrillation patients randomized for an AF ablation or AV node ablation. We have ECG-gated CT for these patients, which we can use to build our anatomical model. But we also have a lot of intraprocedural data with atrial maps and pressure measurements in all 4 chambers. And importantly, we acquire these intraprocedural measurements for baseline and at least 2 different pacing combinations and at 2 different pacing rates. And the plan is to use a subset of these measurements to apply this fitting procedure and create a patient-specific model, and then use the remaining measurements to validate the model, to provide kind of confidence that the model is doing what it is expected to do. So with this, I would like to conclude my talk. So I've shown you that we can use multi-scale electromechanics model to link different processes of cardiac function to clinically measurable outputs. And I've also shown you how we can use emulators, sensitivity analysis, and history matching to iteratively get close to the target values for different outputs of the model. With this, I'm happy to take any questions.
Thanks for the lovely talk. Did I get it right that as you were shrinking down the non-implausible space, you redid the global sensitivity analysis and the most important parameters were changed?
Yeah.
That's really nice, sort of underappreciated thing, I think, that a lot of people would do one sensitivity analysis and then you don't, but how that changed was quite interesting.
Yeah, so what we realized after a lot of iterations of this and trying to do the parameter inferences in a consistent way, because what we want to do is to do this for many hearts. So we need a consistent and standardized process, right? So what we realized was that if we take the important parameters, the first ones affect everything. And as you get rid of them, you get into parameters that affect just one output. So then the fitting gets better. And only with the first GSA, we were able to get only that close to the target values. So that was quite a nice iterative procedure, I think.
And you end up with the same parameters on different patients.
Yeah, we will see how that goes. Yeah.
Yeah, lovely. So how stable are these results to your initial choice of parameter ranges?
The parameter range we chose at the beginning are huge. So hopefully, for two reasons. The first one is that if we were not generous at the beginning, we might have needed to redo the GSA again, which is very expensive, so that is not ideal. And the other reason is that we want to apply this for different hearts, so hopefully the generous ranges at the beginning allow us to be consistent for different patients.
Can I have one more?
Very quick.
Very quick. So how do you then, so in the initial phase, how do you then restrict? You've got enormous parameter ranges. How do you then restrict the effects that are contributing to non-physiological outputs? Because if you've got a parameter that moves in a certain direction that produces a ventricular pressure, maximum pressure of 20, yeah, that's really influential, but you're not necessarily interested in that.
But that's why we do it iteratively. Because in the first iteration, yeah, you have the unphysiological outputs, but then you select important parameters, and so you get more you move more into a physiological space, and then you redo the GSA, and you kind of got rid of, the bad dynamics, basically.
Well, thank you, and thank you to all the speakers this morning.